Okay. Thank you for the opportunity to speak today. I am an associate staff scientist at the Stanford Synchrotron Radiation Light Source, and we just heard from Riti a few talks ago. I work closely with Riti within this structural and molecular biology outreach program that we have, and this project that I'm going to talk today is a result of that outreach. And so yesterday we heard a lot about some essential nutrients, and we heard from Jade about nitrogen. And potassium is one of those key nutrients for plants to be healthy and to grow. And so potassium is associated with the movement of water, nutrients, and carbohydrates through plant tissue. It's important to enzyme activation, such as ATP in plants. It's actually been shown to help with mitigating drought stress tolerance within various plant and microbial species. And importantly, potassium for us actually helps to maintain our fluid balance within our cells. And we get a lot of our potassium from eating fruits and vegetables. And so the diagram that I have here is actually just a kind of simplified version of the potassium cycle with a focus on soil potassium cycling. And you can see that there are various inputs and outputs to the system. And one thing that was brought up yesterday was that we need sustainable alternative pathways to being able to increase the bioavailability of nutrients to plants from soil without the use of fertilizers. And potassium fertilizers aren't necessarily limited, but the mining of potassium fertilizers actually contributes significantly to food-associated greenhouse gas emissions. And so what you can actually see from this diagram, though, is that there are various pools of potassium within the soil. And in fact, soils can actually contain up to 3 weight % potassium. The problem here is that a lot of this potassium is actually within this mineral structural potassium. So that is potassium that is structurally within minerals like potassium feldspar. And in some cases, that can actually be 98% of that soil potassium is within those soil minerals. And so the inorganic weathering of these minerals is far too slow with respect to growing seasons. Inorganic weathering of these minerals can be on the orders of hundreds to tens of thousands of years, depending on various environmental conditions and crystal size. One potential solution is potassium solubilizing microbes. And these are microbes that are known to be able to weather potassium-rich minerals in order to uptake potassium directly from them. Again, there are a couple of caveats or issues with this, is that some potassium solubilizing microbes don't actually always have a positive interaction with plants. And of the available studies that there are, they have relatively limited spatial and or temporal resolution. And lastly, and perhaps most importantly to this talk, is that there are very few capabilities that exist that allow us to actually visualize potassium transformations. And so the molecular mechanisms of potassium translocation from minerals by microbes and to plants is not well understood. And so we are hoping a potential solution to this is potassium synchrotron XRF imaging and potassium thing spectroscopy that we heard a little bit about from Riti. So hopefully this will play. For those of you who haven't done synchrotron XRF imaging before, this is a very short schematic that essentially shows that your sample rasters within the X-ray beam. We can't move X-rays. So this is our incident X-ray beam in yellow at a particular energy that we've selected based on your element of interest. And then over here, this is our detector. And you might be able to tell that there's actually different colors fluorescing from our sample. And our detectors are also energy sensitive. So you can actually map multiple elements at one time, and we can discriminate which elements those are. The result is something like this. We've seen many different types of maps over the past couple of days. Whenever I show you a map like this, warmer colors correspond to higher abundance and cooler colors correspond to lower abundance. This is a map of total sulfur within a moss leaf. And so you might be thinking, well, how does this differ to other elemental imaging techniques? And that's based on the fact that we have monochromatic X-rays. And so if I go to a single spot within this map, a single pixel, we can actually obtain a microZanes spectrum from that specific spot on the sample. And so instead of our sample moving, our sample stays fixed so that the incident X-ray beam can hit the spot that we're interested in. And our incident X-ray beam actually changes in energy, going from an energy that's below the edge of the element of interest to above, generating a Zane spectrum. And when we combine these techniques together, we can do this type of chemical imaging. We've heard also a lot of people mention chemical imaging, and the type of chemical imaging I'm specifically going to be talking about is chemistries of a specific element. And so this example here is showing you a tricolor map of this exact same moss leaf, showing reduced sulfur in red, sulfonate in green, and sulfate in blue. And this type of chemical imaging can be applied to many different elements. Like Riti mentioned, we can do this from phosphorus through a majority of the rest of the elements in the periodic table. And this type of chemical imaging is very good in instances where you actually have a variety of different oxidation states expected in your sample. And so this is what a beamline in the hutch looks like, again, for those of you that have never been. And so this is actually where our X-rays enter our chamber. This case, in this beamline, our chamber is just a helium purge chamber so that you can reduce X-ray attenuation in air. This is our focusing optic. And so with µ-XRF imaging, we have the ability of having an X-ray beam spot size of 0.5 micron. We can load multiple samples at one time. And then over there is our detector. Something that might be of interest to people in the room is that we recently installed this copper braid. This has the ability of keeping your sample at about 0 degrees Celsius, so you can actually keep it just about frozen. Okay, so potassium XRF imaging and Zane spectroscopy. So this is just a quick search I did on Web of Science, looking at where I searched for potassium Zanes in the title of publications versus phosphorus Zanes. So potassium publications are shown in red, going back about 20 years. And so I wanted to show you this because there are actually quite a lot of people that have published work using phosphorus Zane spectroscopy in environmental science. And we can see that comparatively that there is much less literature available for potassium Zanes. And so that's why we're really doing the theory work that Riti mentioned. And so just to reorient you, this is an example of an X-ray absorption spectrum. And like Riti mentioned earlier, that we have these two regions, Zanes and XFs. We're really going to focus on the Zanes region that can give you electronic and geometric information. And again, importantly, oxidation state. And so one of the reasons I chose to show you phosphorus here in this plot is because, like I mentioned, there's a lot of literature in environmental phosphorus Zane spectroscopy. But what we can see here is actually a lot of these Zane spectra are very similar. And that's because the predominant form of phosphorus in the environment is phosphate. And so more recently, there are a handful of groups that have been looking at potassium Zane spectroscopy with applications to environmental science. And like Riti mentioned, this Zanes region is much more rich than we had previously anticipated. And so this is why we have been working with folks at PNNL and EMSL to really understand what it is that's giving rise to these very unique features that we're seeing. Okay, so now to actually show you some of the exciting research. So bringing you back to my example of the potassium cycle, again, we're really interested in how potassium is being uptaken from mineral surfaces by microbes into plants. And so I've really been working a lot with Arunima in this, as well as Jade more recently, where we've been using multiple different types of their terraforms. So this top one is the one that we've been using the most, which I'm going to focus on initially. This is a mineral doped micromodel, or I'll just call it a micromodel. And then this is a schematic of a rhizochip that I'll briefly mention at the very end. So one of the reasons that we've been using these terraforms is because soil heterogeneity and opacity is a central challenge in being able to visualize biogeochemical mechanisms in the soil. And so this micromodel is made of PDMS. You can embed minerals within the PDMS. The gray circles are posts, and so in between the posts is meant to simulate porosity. And then we have 2 nutrient points in the kind of green teal color. And so in this case, we're actually looking at this saprotrophic fungus, Fusarium, within these mineral dope micromodels. And what Arunima and others at EMSL saw was that the fungal hyphae could only bridge to the secondary nutrient port when minerals were embedded within these devices. And they also saw that potassium was actually uptaken within the hyphae using nanosims. And so the questions here then became, what mechanisms did the fungal hyphae actually obtain the potassium? And so that's why we turned to XRF imaging and Zane spectroscopy at SSRL. And so you've actually seen very similar versions of this map in a few talks. This is a map of total potassium within hyphae that were grown on those micromodels. And then we actually removed the hyphae with tape. We can do our microzanes at 2 different spots on the sample. And what we can see here, again, that Riti showed, is that we have this phase of potassium that right now we're calling it organic potassium. We actually don't know how this one is binding. And we suspect it's a mixture. And then this one here is that potassium citrate. We can actually map where these 2 phases occur along the hyphae. And so we've actually seen both citrate and tartrate as hotspots along these hyphae that have been grown in the micromodels. And so we recently looked at a control. So this is just the fungal strain grown on agar with no minerals. And these aren't actually maps of total potassium. These are fitted maps of that organic potassium. So there are actually no other phases of potassium chemistry when this particular fungal strain is grown on agar without minerals. And so this is what all of those microzanes spectra look like from all across the fungal hyphae that are grown on the agar. And so I don't really need to give much of an introduction because we heard a lot about MALDI mass spectrometry imaging yesterday, but we've done some MALDI mass spectrometry imaging of these micromodels, comparing a 7-day experiment and a 30-day experiment. And what you can really see in this 30-day experiment is that the tartaric acid is spatially located differently across the microfluidic device compared to things like citric, fumaric, and malic acids. And so this is actually to do with the biosynthesis pathway of these organic acids as a result of distance from the hyphae to a carbon source. And so subsequent to that, we then had this question, well, what effect is this organic acid, or are these organic acids having on the minerals that we've embedded in the microfluidic device? And so this is one of the experiments, and these red boxes are areas that I performed XRF imaging. In this case, we actually removed all of the fungal hyphae so that we can really be focusing on those potassium minerals that were embedded. And so B and E just correspond to these 2 locations. The tricolor map that I'm showing is 3 different potassium-rich minerals that were embedded at the start of the experiment. C and F are those exact same locations as B and E, but now in blue, we actually see the formation of a montmorillonite clay that has potassium in the interlayer. And so what we've observed here is that we're moving potassium from a non-bioavailable to an exchangeable potassium pool. And so that's a result of the organic acids that are weathering those minerals. And so just as a final example, recently we have looked at a rhizochip with a brachypodium grown within the rhizochip. And we saw some images yesterday. This is what the rhizochips look like. And when they're doped with minerals, this is what they look like. And this was the sample that was sent to me a few weeks ago. We can do large format imaging of at SSRL. And so this is just an example. I'm showing you the silicon map because you can really see the nice pore spaces within the rhizochip. And then this is the map of total potassium. And so we do have the ability to do large format imaging at the various beamlines that we have at SSRL. And then we can move to doing higher resolution imaging of the same sample. And so we actually think over here that this is a sneaky fungal spore that got into this sample that wasn't meant to be there. And so we're interested here also in what the plant is doing to the minerals, if anything. And so in this case, I actually removed the root and I imaged the rhizochip beneath where this root was. And just so very, very quickly to show you here, these are the minerals that are embedded, showing the total potassium abundance. And this is the exact same area shown below. The primary minerals that were embedded at the start of the experiment, secondary being minerals that have formed as a result of the experiment. We can actually see that there's a lot more diffuse secondary minerals that have formed in comparison to the micromodel I showed a few slides ago. And so I guess this has developed a few more questions that we're going to be working on through our collaboration. And one last thing I want to show you is that we've actually also done this XRF imaging on the brachypodium and the brachypodium root hairs. And we can actually see here that there are hotspots of potassium nitrate associated with these root hairs, as well as potassium phosphate. In addition, we've seen more abundance of potassium oxalate and potassium acetate. And so our question here is now, is the presence of potassium nitrate specific to plants? And I will say that I have also recently looked at an ectomycorrhizal fungi, which also did show this potassium nitrate species. And so before I finish up, I want to have a small caveat or word of caution that applying this type of potassium Zane spectroscopy very broadly to environmental samples is still going to be incredibly difficult. And so these two data sets are actually showing bulk potassium Zane spectra, so that's where you homogenize some of your sample. And we actually can see that they're likely losing some of their information by doing bulk, but we actually also don't really have very good standards for being able to interpret these spectra very well. And I'll continue my words of caution that these spectra are microzanes that were from that pesky fungal spore that got into the rhizochip. And I have no standard materials that have been published in the literature or ones that we've obtained that match any of these spectra. And so we really have to continue with this XAS in theory that Riti mentioned. And I think I'm probably out of time, but we have a variety of different conclusions. We have a lot of work to do going forward. We're going to submit a large scale proposal for this and additional theory work in the next call. And like Riti mentioned, we have this small but growing team of outreach at SSRL, and we're available to help you form your experiment. And we really do a lot of research within the plant rhizosphere realm. And so with that, I would like to acknowledge everyone that has made this research possible, and please feel free to email us if you have any questions.
Thank you, Jocelyn. So we have some time for questions if you have any.
Yeah, thank you. That was a great talk. My name is Devin Ripner. A quick question for you: how big are for the imaging data that you're doing, where you have the spectroscopy for each pixel? How big is the data, and how long does it take to collect?
Oh yeah, so a lot of the potassium maps that I was showing, especially where I was showing where the differences in the potassium chemistry is, those maps were typically mapping on the order of millimeters. And because you have to map at multiple energies, as opposed to taking an actual Zane spectrum per pixel, it can take anywhere from 3 to 8 hours, depending on the resolution you want, the size of the map, the number of energies, so that you can build those chemical maps. So yeah, it can take some time. It's not lending itself quite at the moment to like dynamic studies, but that's a future hope.
Okay, and sorry, last question: how big would a data cube be coming out of this?
Ooh, I mean, it depends. How long is your beam time? How many samples do you have? I mean, the amount of data that you collect during beam time can easily be stored and processed on a regular laptop. If you have lots of large format imaging data, typically tends to take longer. They're much, much larger. But we also do this thing where we store some of your data in a completely separate, very large zipped file that you can really only access if you absolutely need it, but we store that all separately because it would just be unmanageable otherwise.
Perfect, thank you.
Okay.
Jocelyn, it was a wonderful talk. This is me just trying to learn from what you guys are doing here. Somewhere around slide 225 or 226, I'm always off by a few because I write the question down and then
Okay.
You mentioned that You saw that, I think that the potassium was going to a montmorillonite clay.
Yeah.
What was how did you know it was going to the montmorillonite clay? Was there something was it a spectral,
Yeah, it's changes in the
spectroscopy from
spectroscopy.
potassium? Or
Yeah.
was it a diffraction scan?
No, it's changes in the spectroscopy. Yeah. So we have a variety of different potassium zanes from minerals, where we have our standards, and there's a lot in the literature. What we're seeing for like the montmorillonite clays is there's quite a broadening in the zane spectra, as well as essentially no post-edge features. And so
It's coming from the potassium zinc.
it's,
Potassium zanes is what's telling you that that's
yes,
clay there.
potassium zanes, yes. And when we combine that with the multi-energy mapping, that's how we can map out the differences and where they exist across the sample.
Thanks.
Okay.
Okay, one last question.
In your earlier results, you were showing contrasts of different dicarboxylic acids, fumarate, tartrate, etc. And I think you were just starting to allude to maybe acetate. Are you going to do a series of the monocarboxylic acids, propionate, butyrate, etc.? And what do you anticipate there? Because those are, of course, microbially active.
That's a good
metabolites.
question. I would say at the moment, we don't necessarily have anything planned, but our future goal is to continue combining these multimodal imaging techniques with the MALDI at EMSL and imaging at SSRL. And there's actually a lot more MALDI data than I showed, which came out in a paper very recently that Arunima wrote, which is very nice. So there could be a lot of data already in there that exists that I just didn't show. But yes, the goal is to continue doing this in the future, this multimodal imaging. And we have some software now that has been developed at SSRL, which should be able to read in the XRF, the MALDI, SEM images so that you can actually process different types of imaging data in one platform.